Now let's talk about transcription and translation. Though before we get too much into that, I do want to make sure that I just double check we're on the same page with what's going on here. So replication is one thing. It is distinct from transcription translation, and it is when we need to make new cells. This happens when you need to make new cells. Let me see if I can write that a little more clearly. This happens when we need to make new cells, and therefore we need to copy the genes. Transcription and translation happen when we need proteins. And frankly, in a cell's life, these are happening at almost completely different times. So replication is a process that happens when a cell is replicating. Transcription and translation is what happens to the DNA the rest of the time. They don't happen in an order. There's no need for replication before transcription or translation. You can do transcription or translation with any DNA that you have. You just do it with the DNA that you need in order to make the specific protein that you're looking for. And where are your genes, anyway? So there is going to be a chromosome, something that has 10 to the 7th to 10 to the 10th base pairs. So we're talking millions to billions of base pairs long. Any individual gene is between 1,000 and 1,000,000 genes long, and usually much more at the lower end of that. And so you only need a tiny chunk of DNA to get transcribed, relatively speaking, in order to get the protein that you want, in order to get the function that you want. So what do you do? How do you find the appropriate gene? Remember, transcription, we're using DNA as our template, as the thing that we're Xeroxing, in order to make RNA. And this is happening in animal cells in the nucleus. So there's a piece of DNA, and then this piece of DNA has a signal on it that's called a promoter. A promoter is essentially a DNA sequence that says, yes, this thing here, this is the thing that we care about, this is the thing that we want. And so when a promoter is found, the DNA is going to get split open like this. So we took this top strand, and I apologize, it's a little janky because we're using text to represent something that's an actual physical thing, but doing our best. It gets split open. Don't worry about these 5' and 3'. This is the nucleic acid chemistry that we're not really digging all the way into. But it gets split open, and then an enzyme called RNA polymerase, so RNA for RNA, ribonucleic acid, our RNA pol is going to sit right here on the promoter, and its job, the thing that it does, is it makes an RNA polymer. It makes a long string of ribonucleic acid based on the same sort of chemistry that we used for DNA replication already. The one big difference between DNA and RNA is now the pairing is A with U and C with G. U takes the place of T's in RNAs. It's just how RNA works. And so I should probably do that in the appropriate color, shouldn't I? A with U. No. A with U. C with G. So wherever there is a C in the DNA, there is a G in the RNA, and vice versa. Wherever there's an A in the DNA, there's a U in the RNA, and so on and so forth. That means that this RNA strand here in blue is almost identical to the one above. The only difference is that substitution of U's for T's. So now we've made our copy. We've made our scribbled down little recipe. It has exactly the same information, but it's a copy. It's a disposable little thing that we don't really care about as much. After transcription, though, while we have our messenger RNA, our disposable copy of the RNA, shall we say, we haven't made the protein yet. That's the next step. That's what's called translation. And just as an aside, something that may help you keep these straight, what does a court transcriptionist or a medical transcriptionist do? They take everything that's said in a certain context and they write it down exactly as it was said, with no changes, just changing spoken English to written English, or spoken Farsi to written Farsi, or spoken Spanish to written Spanish, or whatever. What does a translator do? A translator takes information in one language and translates it to another. So going from written English to written Farsi, or written Spanish to written Japanese. In transcription, it is an absolute perfect, or as perfect as humans can ever manage, one-to-one transfer of information. Lots of times with good transcription, they have all sorts of things that they use to indicate pauses and ums and hms, so you don't really lose any information other than, I guess, inflection perhaps. With translation, those of you who speak more than one language know it's not exactly one-to-one. And so, while you're still going to have the heart of the information there, the poem that is translated is still going to have the same overall meaning and sense. It's not exactly the same, because you are switching from one language, nucleic acids, to a different one, proteins. Transcription is DNA to RNA happening in the nucleus. Translation is RNA to protein, and that is happening out in the cytosol. So, translation, how does that happen? Okay, there's a lot to this, and I am going to be skating over a whole heck of a lot of details here. And frankly, this particular diagram is a bit busy. There's some videos that I'm going to link from the DNA Learning Center that I think help out with this a bit. The kind of instead of static images, it's dynamic video. It really helps with seeing what's going on. But here's the big thing. We have our RNA that we transcribed. And we're going to go from an RNA language to a protein language. How the heck do we do that? The big important things here are that the ribosome uses these adapter molecules called tRNAs, or transfer RNAs. A tRNA has on it a series of 3 bases, or an anticodon, that's able to bind with 3 bases that's on the RNA. At the other end of this, we have an amino acid. Next to that, another tRNA is going to bind to its specific 3 nucleotides, and it's going to be carrying a different amino acid. The ribosome, this really interesting and very complicated piece of molecular machinery that's made of RNA and proteins, it is going to hook those 2 individual amino acids together, and as the ribosome moves down, as it goes along this RNA, tRNAs are going to bind at 3 and 3 and 3, carrying their amino acids, which are all going to get hooked together, one right after the other. making that protein. Which if you go back to when we were talking nutrition and proteins, series of amino acids like beads on a string. This is the really big overview. We're not going to get too far into the details, but this is the big overview of what happens. What do I want you to get about this? Well, again, we're not doing replication, we're doing transcription and translation. And actually, let me draw that a little bit differently because DNA is involved at the beginning of transcription. So, transcription is the process of going from Grandma's old family super precious recipe book to a disposable copy of just the page that you need, not the whole thing, just the page that you need, that has nonetheless the information that you're looking for. In order for this to happen correctly, a lot of proteins have to be in the same place at the same time, working together. Once that RNA has been made, 3 nucleotide chunks are going to specify individual amino acids in the protein, and this is all put together, oops, not quite right there, and this is all put together by the ribosome. And so we have to have another set of players that are all working together in order to change from nucleic acid language, which is just the 4 bases, to protein language, which is the 20 plus amino acids. And that is, in a slightly cramped nutshell, how we get from a gene to a protein, which is going to get us to a